Okay, so my presentation is over RT-PCR. Some background and principles. RT-PCR is a lab technique that combines the reverse transcription of RNA into DNA and amplification of certain DNA using the polymerase chain reaction. RT-PCR is mainly used in order to measure a certain amount of RNA. They do this by monitoring the amplification of the fluorescence on that particular sequence. RT-PCR has revolutionized the study of gene expression in several ways. One is being able to detect the transcripts of many genes. It has also enabled sample amplification. So there is also one-step RT-PCR and there's two-step RT-PCR. One-step RT-PCR is going to use a single tube for the entire reaction from the cDNA synthesis to PCR amplification. So you can see in this figure up here, it's going to have both the one step at the top and two step at the bottom. So for one step, the RNA, the primers, the reverse transcriptase, DNA polymerase, and the buffer reagents are all going to be placed into the first single tube. They then are going to cycle this tube, and the first cycle that they do is for the reverse transcription. They'll then run, I believe it's about 30 more cycles, and that's going to denature the cDNA. The PCR is then placed in, in gel electrophoresis. That way they can analyze it. So for the second one, it's a very similar process, except we're going to use two separate tubes. They will place the RNA, a non-specific primer, reverse transcriptase, DNA polymerase, buffer reagents, and they're going to put that in a single tube to begin with. They then are going to cycle that tube for one time. After it's cycled one time, that's when the reverse transcription is going to take place. So we immediately need to either put it on ice or take it to run the PCR. So then we're going to add a specific primer and run the PCR in a separate tube. General applications of techniques. RT-PCR is used to detect genetic diseases as well as measure gene expression. It's been used for several research methods to measure gene expression as well. RT-PCR, which is real-time, was used to measure gene expressions of gal genes in yeast cells. This is an example of how it's been used before. Gene insertion is also another thing that it's being used for. So they have used it to insert eukaryote genes into prokaryotes. This is because the cDNA is generated by the RT-PCR is the same sequence that would ultimately be translated into the protein after transcription if that was going to take place. Scientists are also working with it to see if they can use it for cancer detection. They're working to use this to analyze the expression levels of certain types of cancer cells that are present. So you can see in this figure, they basically are going to take his condensation from his exhaled breath. They're going to add it to a PCR once they add all the reagents, buffers. They run the PCR and they're able to see if he hits in any of the lung cancer stages. Okay, so this was an article I found. In this article, they used the TACMAN probe to determine if there were certain sequences in that gene, and then they ended up using it on cytokine genes expression. So in this figure, the author is asking exactly how the TACMAN probe operates. So we have two parts of the figure, figure A and B. So initially the PCR is set up The PCR will then anneal to the target sequence between the forward and reverse primers. So you can see the probe is right here, forward primer, reverse primer. We have I believe it is a fluorochrome, and then the quencher dye is on this side with the Q. So you can see that in figure B, the probe is basically going to degenerate. So in figure B, we can see that during this extension phase of the reaction, the probe is cleaved, allowing there to be an increase in reporter fluorescence emissions. That way, we can see it on the PCR with the fluorescence. Figure A shows that the fluorescence will not occur if the probe is still intact, meaning that the extension phase absolutely must occur in order for there to be a fluorescence emission for us to see on our PCR. This article basically just shows very well how the TACMAN probe works for PCR. It's actually used very widely now. They then go on in this study to use the method to use this as a method for quantification of cytokine gene expression. The rest of the article is actually really interesting. I'm gonna add the link if y'all wanna go look at it. So my topic related question is that I had previously stated in another slide how scientists are working to use RT-PCR in cancer detection due to tumor cells producing unique mRNA sequences. I'm just wondering if anybody thinks that this will be effective for every type of cancer. And then I have my references. So if anybody has any questions, feel free to leave them in the discussion board for me. And I look forward to hearing what y'all have to say.